This video will demonstrate the DNA Ploidy procedure. Referring to the tumor area marked by a pathologist on an HE-stained section, the corresponding tumor area of the paraffin-embedded tissue is outlined with a needle, and the block is cut in 50-micron thick sections. Preparation of the tissue section starts with deparaffinization using xylol. This is done twice. The tissue is then rehydrated by adding ethanol of decreasing concentrations. During the rehydration process, the tissue is transferred to conical centrifuge tubes. After rehydration, the tissue is washed using PBS. The enzyme protease is then added to the tissue. The tubes are placed in a water bath shaker at 37 degrees Celsius and 200 revolutions per minute for 60 to 75 minutes, depending on the origin of the tissue. The enzymatic activity is stopped by placing the tubes on ice and adding ice-cold PBS. The resulting solution is lightly pumped with a pipette to dissolve the tissue and release singular nuclei. To make the nuclei solution as pure as possible, it is filtered through a 60-micron nylon filter. It is then washed twice by spinning the test tubes in a centrifuge, removing the supernatant with a pipette and adding new PBS. After the last spin, the nuclei are visible as a pellet at the bottom of the test tube. This pellet is dissolved using PBS and is now prepared for cytospin. Cytospin funnel, filter, and slide are assembled for the making of a monolayer. Calculated amounts of suspension are added to the cytospin funnel. After spinning, the monolayer is visible on the slide. This process is repeated with adjusted amounts of suspension until optimal nuclei density is achieved. This is monitored using a microscope. The monolayers are fixed in formalin overnight before the Foilgen staining. This process starts with hydrolysis using hydrochloric acid. The monolayers are stained using the Schiff reagent for 2 hours in dark conditions. Then, the monolayers are washed 3 times with a sodium sulfite solution before dehydration. This is done from H2O with increasing concentrations of ethanol. Finally, the slides are dipped in xylol before they're mounted with cover glasses. Measurements of the monolayers are done using the method image cytometry. The slides are scanned using a microscope fitted with a digital camera. Specially developed software is used to select, segment, and scan singular nuclei. The measurement is based on digital images of the nuclei consisting of pixels with varying grayscale values. These values are converted to integrated optical densities which equals the amount of DNA in each nucleus. The files containing image galleries are then edited using special software. The resulting histograms are analyzed and classified, and the attending physician is informed of the classifications.